In 1993, a physicist named Michael Land sat down to calculate exactly how good the human eye actually is. Land was one of the world's leading experts on animal vision, a researcher at the University of Sussex who had spent decades studying eyes across the animal kingdom. From jumping spiders to scallops to mantis shrimp, he had looked at more kinds of eyes than almost anyone alive. And when he turned that expertise toward the human eye specifically, treating it the way an engineer would treat any optical instrument, what he found was not what most people expect. The cells that actually detect light in your eye face the wrong direction. They point toward the back of the eye, away from incoming light. Every photon that enters your eye has to pass through multiple layers of nerve tissue and blood vessels before it reaches the cells designed to catch it. Any engineer designing an optical sensor from scratch would consider this a fundamental error. The cable that carries visual signals from the eye to the brain does not exit cleanly from behind the retina. It punches through the retina itself, creating a hole, a blind spot, a region in your visual field right now where you are receiving zero information. And you have almost certainly never noticed it because your brain fills it in so completely and so automatically that the gap is invisible from the inside. The sharpness you think you are seeing across your entire visual field right now, the sense of a rich and detailed image stretching from edge to edge, is largely invented. Your eye is genuinely sharp only in a tiny central zone called the fovea, roughly the size of your thumbnail, held at arm's length. Everything outside that zone is low resolution, and your brain assembles the illusion of completeness from rapid eye movements and educated guesses about what should be there. Land knew all of this, and he also knew something that makes the whole picture stranger. The human eye, with all of those apparent flaws, can operate across a range of light intensity spanning 10 orders of magnitude. From starlight to noon sun, it can track a moving object against a complex background while simultaneously reading the emotional state of a face. It responds, under the right conditions, to remarkably small amounts of light. No camera ever built by human hands does all of that. So here is the question that Land kept returning to, and the question that this story is really about. How does the worst designed camera in the engineering world outperform everything we have ever built? The answer is not about engineering. It is about history. 500 million years of it. A history that did not begin with an eye, did not begin with a cell, did not even begin with anything you could see under a microscope. It began with a single molecule in an ancient ocean, a molecule that flinched when light touched it. And from that flinch, everything you have ever seen in your life was eventually made possible. Picture the ocean 600 million years ago. Not dark. That is the first thing to understand. The sun existed. Light poured into the water exactly as it does today. The surface of that ancient sea was bright, and the upper layers were lit with the same kind of clear blue light that fills a swimming pool on a summer afternoon. But nothing alive could use it. Not the way we mean when we say sea. The creatures in that ocean, and there were many of them, could detect chemicals dissolving in the water. They could sense vibration, pressure, temperature, electrical fields. They were sensitive to their environment in dozens of ways we do not fully appreciate. But light, pouring down through the water in quantities that would have been blinding to a modern eye, carried no useful information. It landed on living tissue and did nothing. It was noise, background radiation, a physical fact with no biological meaning. And the consequence of that was a world without distance. Think about what that actually means. every piece of information about the world around you, if you lived in that ocean, arrived by contact. Something touched you, or chemicals from something nearby dissolved into the water and reached you, or the pressure wave from something moving passed through your body. Your entire model of the world extended only as far as your body could directly sense. There was no over there. There was only here and not yet here. A predator approaching you from 3 feet away was, in every functional sense, invisible until the moment it was close enough to touch. The gap between detection and contact was nearly zero. And in that gap, or rather, in the absence of that gap, survival looked very different than it does today. You did not run from threats you could see coming. You reacted to threats that had already arrived. For most of the history of life on Earth, this was the only way things worked. Life emerged around 3.5 billion years ago. For roughly 3 billion of those years, the most sophisticated thing any living creature could do with light was notice that it existed. Single-celled organisms developed patches of pigment that could distinguish brightness from darkness. And this was genuinely useful. A photosynthetic cell that could orient itself towards sunlight gathered more energy. A cell that could detect the sudden shadow of something large passing overhead might have enough warning to sink slightly in the water column. Light as an on-off switch. Light as a rough environmental cue. Light as a signal that day had become night. But not light as information about space. Not light as a map of what was where. There is a single-celled organism called euglena that lives in freshwater ponds all over the world today. It has a small, pigmented spot near one end of its body. And for a long time, biologists called this spot an eye spot and wondered if it was a primitive precursor to vision. In one sense it is. The pigment in that spot shields part of a nearby light-sensitive structure, so that the cell gets slightly different light signals depending on which direction the brightness is coming from. It is a crude direction finder. It tells euglena something about where the light is strongest, and euglena uses that information to swim toward it. But there is no image, no shape, no distance, no object, just a gradient, a difference in light intensity between one side and another, translated into a swimming direction. That is where vision begins, not with seeing, but with a shadow being meaningful. The reason this matters, the reason the gap between that simple light-sensing ability and actual image-forming vision is so important, comes down to one concept that changes everything once you understand it. Speed of information. Chemical signals travel through water at the speed of diffusion. By the time the chemical signature of a predator reaches your senses, that predator is already close. Pressure waves travel faster, but they still require the source to be relatively near. Both of these sensory systems tell you about the present, about what is happening right now in your immediate vicinity. Light travels at 299 million meters per second. When light bounces off an object and reaches your eye, it is carrying information from a distance. Information about something that is out there, away from you, potentially with enough space between you and it to do something about it. The first time any living creature could extract directional information from light, it gained something that no living thing had possessed in 3 billion years of evolution. A warning before the danger arrived. Not a clear one, not a detailed one, just a shadow, a shift in brightness, a signal that something was moving somewhere in that direction. But even that was enough to begin changing the math of survival in that ancient ocean, and something about what came next would change everything else. A photon leaves the surface of the sun. It travels 150 million kilometers through empty space. It takes 8 minutes. When it arrives at Earth, It passes through the atmosphere, through the surface of the ancient ocean, through layers of water, and it strikes a single protein molecule smaller than anything you could see with the naked eye. The molecule bends, not breaks, not dissolves, bends. One small section of it, a chain of carbon atoms arranged in a specific curved shape, straightens out when the photon hits it. The whole event takes 200 femtoseconds. That is 200 quadrillionths of a second. It is the fastest chemical reaction in all of biology. And it happened billions of years before there was any brain to receive the signal it created. That molecule is called opsin. And the story of every eye that has ever existed on this planet begins with what happens in the fraction of a second after it bends. When opsin changes shape, it triggers a cascade. The newly bent protein activates a neighboring molecule, which activates another, which eventually opens or closes tiny gates in the cell membrane, which changes the electrical charge across that membrane, which sends a signal down whatever nervous tissue happens to be connected to it. Light becomes electricity. Electricity becomes information. And information, if there is anything around to use it, becomes behavior. The elegant thing about this system is how little it requires to get started. You do not need an eye. You do not need a retina or a lens or a brain or any of the complex machinery we associate with vision. You need one protein, one molecule of a vitamin A derivative called retinol that nestles inside the opsin like a key in a lock, and enough light to hit it. That is the entire foundation. Everything else that evolution ever built around vision, every compound eye and camera eye and mirror eye and pinhole eye across 500 million years of animal life, is an elaboration on that single molecular event. What makes opsin extraordinary is not just what it does, but how old it is. Proteins that use retinol to respond to light show up across an enormous range of living things. The molecular machinery for turning light into a biological signal appears to have very ancient roots, predating the complex eyes we see in animals today by a vast stretch of time. The specific animal opsins in your eyes and the opsins in the eyes of a jumping spider trace back to a common ancestor, so distant it makes the dinosaurs look recent. And while the light-sensitive proteins found in bacteria and single-celled organisms are related but distinct from animal opsins, the deeper point holds. Retinol-based light sensing is one of the oldest molecular tricks in the history of life. Life found this trick very early and never let it go. In 1995, a geneticist named Walter Gehring and his team at the University of Basel did something that sounds like it belongs in a science fiction story. They took a gene called PAX6, known to be essential for eye development in mice, and they inserted it into fruit flies, not in the head, where eyes normally form. They inserted it in the tissue that would become wings, and legs, and antennae. The flies grew eyes in those places, not mouse eyes, fly eyes, fully formed compound eyes, in the exact structure of a fly eye, sprouting from wings and legs and antennae where no eye had ever grown before. What Gehring had found was something that reshaped how biologists think about the evolution of vision. PAX6 is not a blueprint for a specific kind of eye. It does not carry the design of a fly eye or a mouse eye or a human eye in its sequence. What it carries is a command, a signal so ancient and so conserved across hundreds of millions of years of evolution that it can be read by almost any animal alive today. The command says, "Build an eye here." The animal then builds the eye in its own way, using its own developmental programs. producing whatever kind of eye its lineage has evolved. The blueprint is local. The command is universal. And the fact that a single gene can issue that command across the full distance between a mouse and a fly, two animals whose last common ancestor lived over 500 million years ago, tells you something profound about how deep the roots of vision actually go. Before there was an eye, there was opsin. Before there was a retina or a lens or anything you would recognize as sight, there was a molecule responding to light. And that response, replicated and built upon across billions of years, became the foundation for everything that followed. The molecule flinched, and from that flinch, the next problem became obvious, even if it would take hundreds of millions of years to solve it. Knowing that light exists is one thing. Knowing where it is coming from is something else entirely. Imagine you are a small, soft-bodied creature in the ancient ocean. Around 540 million years ago, you have photoreceptors. Your cells can detect light, respond to it, send signals based on how bright it is. In the slow, chemical world of the Ediacaran period that preceded this moment, that was enough. The creatures around you navigated by touch and taste and the slow drift of molecules through water. Nobody could see you from a distance. Nobody was coming for you from across the water column with deliberate intent. But something has changed. Somewhere nearby, in a lineage you have no awareness of, a creature has developed something new. Not yet a full eye, not yet an image, but something that gives it information your ancestors never had to worry about. It can detect the direction of light. It can sense your shadow as you move above it. And it is starting to move toward that shadow with a purpose that the old world never contained. You are about to become prey to something that can partially see you, and you have no answer for it yet. The physics of that problem has exactly two solutions. Both of them were discovered by evolution, independently, probably multiple times, in the millions of years that followed. And both of them begin with the same basic insight. To get information about direction from light, you have to create shadow. The first solution is the cup. A flat sheet of photoreceptors tells you that light exists, but not where it is coming from. Because photons arrive from every direction simultaneously and hit every cell at once. But if that flat sheet bends into a shallow bowl, something changes. Light coming from the left side of the world strikes the right inner wall of the bowl more strongly than the left. Light from above hits the bottom. Light from below is partly blocked by the rim. The bowl creates shadow, and shadow creates difference. And difference is information. For the first time, a creature does not just know that light exists. It knows, roughly and crudely, which direction the light is coming from, which means it knows, roughly and crudely, where the shadow came from, where the large thing moving overhead came from. This sounds almost too simple to matter. A bent sheet of cells, a bowl. But watch what happens when you make the bowl deeper, when the opening narrows, when the ratio of depth to width increases. The shadow effects become more pronounced. The directional sensitivity sharpens. An animal with a deeper cup knows more precisely where danger is than an animal with a shallower one. And each incremental improvement in that precision is a genuine advantage in a world that is becoming more dangerous by the generation. In 1994, two Swedish researchers named Dan Erik Nilsson and Susanne Pelger built a mathematical model of this process. They calculated how much improvement each small change in eye shape would provide, and how quickly natural selection would favor those improvements. Their conclusion was striking. Under reasonable assumptions, The sequence from flat photoreceptor patch to sophisticated camera eye could happen in less than 500,000 years. In geological time, that is nearly instantaneous. The model describes optical geometry, not the full complexity of building a real eye with all its genes and tissues, but it makes a point worth holding on to. The evolution of the eye does not require a miracle. It requires only that each step be slightly better than the last. And in a world full of things trying to eat you, even a tiny improvement in knowing where they are is enough to change who survives. Now, here is the second solution. While the cup was deepening in some lineages, other lineages were taking a completely different approach. Instead of bending the photoreceptor sheet into a bowl, they closed almost everything off, leaving only a tiny hole for light to enter. This is the pinhole. If you take a dark interior with photoreceptors lining the inside, and you make a small hole in one side, something remarkable happens. Each point in the outside world can only send light through the hole to one specific point on the interior surface. The geometry forces a mapping. Points in space correspond to points on your photoreceptor sheet. You have an image. The animal that exists today, still using this solution, still navigating its world with a pinhole eye, is the nautilus. Its lineage has changed considerably over the vast stretch of time since the early days of complex eyes. But the nautilus preserves an optical approach that was viable long before lenses existed. When you look at a nautilus eye, you are looking at a functional, image-forming eye with no lens at all. Just a small opening, a dark interior, and a sheet of photoreceptors doing the work. The pinhole works. It genuinely produces images. Not blurry gradients, but actual spatial maps of the world. But it carries a brutal trade-off baked into its physics. The smaller the hole, the sharper the image, because less overlap occurs between light from different directions. And the smaller the hole, the less light gets in. Make your pinhole eye sharp enough to see clearly, and you have made it nearly useless in anything less than bright sunlight. Open it up enough to work in dim light, and your image degrades into something closer to a blur. The creature in the ancient ocean that we started with, the one becoming prey to something that can partially see it, is now in an arms race. It did not choose to enter. The cup gave its predators direction. The pinhole gave some lineages, images. But neither solution solved both problems at once. Sharpness and brightness, distance and detail. Every living thing that tried to see the world clearly ran into the same wall. That wall had only one answer, and the answer, when it came, should not have been possible to make from living tissue. Hold a single drop of water up to the light. The drop curves. Light, passing through it, bends toward the center. Converges. Focuses. If you hold it over a piece of text, the letters underneath appear larger. If you hold it in direct sunlight, it concentrates the rays into a point hot enough to burn paper. That drop of water is a lens. It did not need to be designed. It did not need to be manufactured or polished or coated with anything. The physics of refraction, the bending of light as it crosses from one medium into another with a different density, did all the work automatically. Any transparent material with a curved surface does this. It cannot help doing it. And somewhere in the ancient ocean, a cluster of cells in a developing eye happened to accumulate a protein that made them slightly more transparent than the cells around them. The light bent. Not much. Not enough to produce a sharp image. Not enough for any creature alive at that moment to notice the difference in any conscious way. But enough that the animal with those slightly transparent cells gathered fractionally more light onto its photoreceptors than the animals around it. enough that in the relentless arithmetic of survival, where fractional advantages compound across thousands of generations, those cells were worth keeping. That was the beginning of the lens. The proteins that make up the lens of your eye are called crystallins. What makes them remarkable is not what they are, but what they were before they became lenses. Many crystallins are proteins that already existed in cells for completely different purposes. Enzymes, proteins involved in stress responses, molecules that had been doing other jobs for hundreds of millions of years before some of them ended up concentrated in eye tissue and turned out to be transparent enough to bend light usefully. Evolution did not invent a new protein to make lenses. It looked at what was already available and found that some of it happened to be transparent. This is how evolution solves problems in a way that no engineer ever would. An engineer designing a lens starts with the requirement and finds the material. Evolution starts with the materials that happen to exist and finds the requirement they accidentally satisfy. The lens solved both problems that the cup and the pinhole had separately failed to solve. A lens in front of a photoreceptor sheet gathers light from a wide angle, the way an open eye gathers light, but focuses it onto a specific point on the retina, the way a narrow pinhole would. You get brightness and sharpness at the same time. The trade-off that had constrained every previous eye design simply disappeared. But the story of the lens is not complete without trilobites. Trilobites were among the dominant animals of the Cambrian seas. They appear in the fossil record over 500 million years ago and persist for more than 270 million years, longer than the entire reign of the dinosaurs, before vanishing in the Great Extinction at the end of the Permian period. They were arthropods, distant relatives of modern crabs and insects, and they came in an extraordinary range of sizes and shapes. Some were smaller than your fingernail, some were the size of a dinner plate, and some of them built their lenses out of stone. Not organic protein, not transparent tissue, calcium carbonate, calcite, the same mineral that makes up limestone and chalk and the shells of clams. Trilobites with calcite lenses are among the most astonishing animals that have ever existed. Not because calcite is a superior optical material in every way, but because what they did with it reveals something extraordinary about the power of natural selection to find solutions in unexpected places. Calcite has one serious optical problem. It is birefringent, meaning it splits light into two separate beams, traveling at slightly different speeds, which would normally produce a doubled and blurry image. Any optical engineer, handed a piece of calcite and asked to make a lens, would tell you immediately that you cannot build a useful lens from this material without compensating for that splitting. Trilobites compensated for it. In the 1970s, researchers examining trilobite eye fossils under high magnification discovered something that stopped them cold. Some trilobite lenses had a complex internal structure, a secondary lens element nested inside the primary one, with a curved interface between them, designed to correct exactly the aberrations that calcite would otherwise produce. The geometry of those interfaces corresponds to solutions that human mathematicians and optical engineers worked out centuries later. René Descartes described the ideal lens shape for eliminating certain optical aberrations in the 1600s. Christian Huygens worked on related problems in the same era. Some trilobites had already built those solutions into stone eyes hundreds of millions of years earlier, not because they understood optics, because the animals whose eyes happened to have better geometry saw better, found food more effectively, escaped predators more successfully, and left more offspring. Over enough generations, across enough death, the geometry improved. The math emerged from survival, not from calculation. natural selection, given enough time and enough generations, finds things that human intelligence takes centuries to discover. Now here is what the lens made possible that nothing before it had. Every previous eye design, the flat photoreceptor patch, the cup, the pinhole, gave an animal information about its immediate surroundings, about things relatively close, casting enough shadow or producing enough chemical signal to be detectable. The range of useful awareness extended maybe a body length or two in most cases. A lens eye changes that range dramatically. A well-designed lens eye can form a clear image of an object many body lengths away. It can detect the silhouette of a predator before that predator is anywhere near striking distance. It can track the movement of prey from across an open space. For the first time, an animal can know about things that are genuinely far away. And in an ocean where for hundreds of millions of years nothing could see anything from a distance. This was not just an advantage. It was a revolution. Because the moment one animal could see from a distance, every other animal in that ocean had a new problem it had never faced before. It could be seen. Start with the animal that had no defenses. It did not need them. For hundreds of millions of years, in the warm, shallow seas before the Cambrian period, the creatures sharing the ocean with it were slow, soft, and essentially blind to anything beyond arm's reach. The world was full of life, but it was life that found food by drifting into it. Life that encountered danger only when danger was already touching it. In that world, being soft and slow and small was not necessarily fatal. The largest predators had the same limitations as everyone else. They could not see you coming any more than you could see them. The entire ocean operated on the logic of proximity. Survival was mostly a matter of chemistry and chance. This animal, soft-bodied, probably a few centimeters long, leaving no hard shell and therefore almost no fossil record, would have been unremarkable by the standards of that world. It ate. It moved. It reproduced. Its ancestors had been doing these things for an almost incomprehensible stretch of time, and nothing about the world had ever required them to do anything different. And then the world changed. Not overnight, not with any single dramatic moment that could be pointed to on a calendar, but in the fossil record, beginning around 540 million years ago. Something happened that has no real parallel in the entire history of animal life before or since. In a period that is, by geological standards, remarkably short, the ocean filled up with new kinds of bodies: shells, spines, claws, jointed legs, compound eyes, mineralized armor, swimming appendages, grasping structures, mouthparts designed not just to filter organic particles from the water, but to seize and pierce and consume other animals. Paleontologists call this the Cambrian explosion. Though explosion is a word that requires some care, the changes did not happen overnight. Tens of millions of years were involved, and there is growing evidence that much of the preparation for what appears in the Cambrian fossil record was already underway in the Ediacaran period that preceded it. The appearance of sudden dramatic change is partly an artifact of how fossils are preserved. Hard shells and mineralized structures leave records. Soft bodies mostly do not. But something real happened. Something accelerated. And the question of what caused it has occupied paleontologists for generations. Because the answer is almost certainly not simple. Rising oxygen levels in the ocean and atmosphere gave animals the metabolic fuel for more active lifestyles. Changes in ocean chemistry made it easier to build calcium carbonate shells. New genetic tools for building complex body plans became available. Ecological relationships between species began driving each other in feedback loops that had not existed before. And somewhere in that mix, vision arrived. The hypothesis that visual predation played a significant role in the Cambrian explosion was developed most forcefully by a paleontologist named Andrew Parker in the early 2000s. Parker's argument was that the appearance of image-forming eyes in predators created immediate and intense pressure on every prey animal in the ocean. An animal that can see you from a distance changes the entire calculus of survival. Defenses that were adequate against blind predators. Defenses based on being small and slow and chemically unremarkable were suddenly useless against something that could spot you from across the water column and come for you with deliberate intent. Parker's light switch theory has been debated, refined, and challenged in the years since. Most researchers today would say that vision was probably one significant pressure among several, not the single cause of everything that happened. The Cambrian explosion was driven by multiple factors interacting in ways we still do not fully understand. But the core observation is hard to argue with. When a predator can see its prey before touching it, prey needs new defenses. When prey develops new defenses, predators need new tools to overcome them. When predators develop new tools, prey needs to evolve again. The loop, once started, does not stop. Go back to the soft-bodied animal we started this chapter with. In the world it was born into, its greatest risk was bumping into something larger than itself. In the world its grandchildren inherited, something could see it from across the water column and was already moving toward it. Not by accident, not by drift, with intent, with eyes that gathered light and focused it and turned it into a spatial map, precise enough to identify a target and pursue it. The most vivid evidence of what that world looked like comes from a place in the Canadian Rockies called the Burgess Shale, discovered in 1909 by a paleontologist named Charles Walcott, whose horse cracked open a rock on a mountain trail and revealed something extraordinary inside. The Burgess Shale is a deposit of mid-Cambrian age that preserved not just shells and hard parts, but the soft tissues of animals that would otherwise have left no trace at all. What Walcott found, and what generations of researchers have continued excavating and analyzing in the more than a century since, is a snapshot of a world in the middle of reinventing itself. Animals with 5 eyes and a grasping proboscis. Animals with circular mouths lined with plates. Animals built on body plans so strange that when the paleontologist Stephen Jay Gould described them in his 1989 book, Wonderful Life, he argued they represented entire branches of the animal kingdom that had gone extinct and left no descendants. Later, research revised some of those interpretations, finding that many Burgess Shale animals were stranger-looking relatives of groups that still exist today. But the basic picture holds. The Cambrian Ocean was a place of radical and rapid experimentation, and much of that experimentation was directly related to the twin pressures of seeing and being seen. Among the Burgess Shale animals, Anomalocaris stands out. It was large by Cambrian standards, potentially reaching 1/2 meter or more in length, at a time when most animals were measured in millimeters or centimeters. It had a circular plated mouth. It had 2 large grasping appendages at the front of its head, and its eyes, preserved in exquisite detail in several fossils, were enormous compound structures. Analysis of Anomalocaris eye fossils, published in 2011, led by researchers John Patterson and Michael Lee, concluded that some specimens preserved surfaces with at least 16,000 lenses in a single eye, giving it visual acuity comparable to many modern predatory arthropods. It could form detailed images, It could track movement. It could almost certainly identify prey from a distance that would have been effectively infinite by the standards of every predator that had existed before it. In the world before vision, the soft-bodied animal we started with had a chance. A blind predator had to find it by chemistry, by pressure waves, by blundering into it. There was reaction time. There was distance that provided a margin. In the world Anomalocaris inhabited, that margin was gone. Some lineages developed shells thick enough to resist crushing. Some developed spines that made them painful to swallow. Some developed speed, or camouflage, or the ability to burrow rapidly into sediment when a shadow appeared above them. Some developed their own eyes, so that the warning system predators had could be turned back on them. And some, unable to adapt fast enough, simply disappeared. What emerged from this period was a world that looks, in its basic ecological structure, recognizable to us today. Predators and prey, active hunters and animals with defenses. A food web driven by visible, directed, intentional feeding rather than by chemical gradients and accidental contact. The world we live in is the world the Cambrian built. The template was set 500 million years ago, in those warm, shallow seas, under the pressure of being seen. But while all of that drama was unfolding in the open water, something else was happening. Some of it was being recorded, not in behavior, not in relationships between species, but in something harder and more permanent, in stone. Pick up a trilobite fossil sometime. If you ever get the chance, you can find them in rock shops, natural history museums, estate sales, the collections of amateur fossil hunters who spend their weekends in the limestone hills of Ohio or the shale beds of Utah. They range from the size of a thumbnail to the size of a dinner plate. And they range in quality from rough outlines pressed into mudstone to specimens so perfectly preserved that you can see individual segments, individual spines, individual facets of the eyes. Look at the eyes. This is not a figure of speech or a poetic instruction. The eyes of many trilobite fossils are physically present, intact, preserved in the rock in a way that soft tissue never is. The reason is the same reason trilobites were so successful for so long. Their eyes were made of calcite, the same mineral as the surrounding rock. And when the animal died and was buried, and the soft tissues decomposed, and the organic molecules slowly replaced themselves with minerals over millions of years, the calcite lenses remained. They did not need to mineralize, because they were already mineral. What you are holding, when you hold a trilobite fossil with intact eye structure, is not a representation of an eye, or an impression of an eye, or a chemical ghost of an eye. You are holding an optical instrument. Lenses that once gathered and focused light, now dark and cold and silent, but geometrically intact. Curved surfaces that, if light were to pass through them at the right angle, would still bend it. The eye itself is there. It just stopped working 250 million years ago. Researchers who study trilobite eyes in detail, using electron microscopes and computed tomography scans and optical modeling software, find things that continue to surprise them even now. The diversity of eye designs across the more than 20,000 known trilobite species spans almost everything that compound eye evolution could produce. There are holochrome eyes, where thousands of small hexagonal lenses pack together tightly under a single, continuous outer layer, producing wide-field coverage with the kind of dense sampling that would give an excellent sense of movement across a large visual field. There are schizochroal eyes, found only in a particular group called the Phacopoda, where larger lenses are separated from each other by thick borders of non-transparent tissue, each lens sitting in its own individual chamber. Some trilobites had eyes mounted on stalks, allowing them to see in nearly every direction simultaneously, while the rest of their body remained pressed against the seafloor. Some had eyes wrapped around their head in a geometry that would have provided nearly spherical coverage. Some, living in deep water where light barely penetrated, had enormous eyes that covered most of the surface of their head, maximizing the collection of whatever faint illumination existed. And some, living in environments where eyes were simply not useful, had no eyes at all. 270 million years of trilobites meant 270 million years of eye evolution, experiment, refinement, and adaptation to every visual environment that Paleozoic seas could offer. The lenses that stopped the researchers cold, though, were the ones in certain phacopoid trilobites. When optical physicist Johan Clarkson and physicist Riccardo Levi-Setti analyzed some of these lenses in detail in the 1970s, they found something that should not have been there. The lenses were corrected. Calcite is birefringent. It splits light. Any simple calcite lens would produce a blurred, doubled image because light traveling through the crystal separates into two components, moving at slightly different speeds. This is a fundamental physical property of the material, not something that can be wished away. But inside some trilobite lenses, the researchers found a secondary element, a precisely curved interface between two regions of slightly different calcite crystalline orientation, positioned and shaped to counteract exactly the birefringence of the outer lens. The geometry of these correction structures corresponds to solutions that human mathematicians arrived at independently. Centuries after the trilobites had gone extinct, natural selection, given enough time and enough generations and enough death, finds things that human intelligence takes centuries to discover. But here is what Clarkson and Levi-Setti could not find, and what no researcher since has been able to find, because the fossil record simply does not preserve it. They could not find what it was like to be a trilobite looking through those lenses. The geometry is there. The optics can be modeled. We can calculate, with reasonable confidence, what kind of image a trilobite eye would have formed. We know roughly what wavelengths of light the calcite would have transmitted best. We know the angular resolution, the approximate sharpness, the field of view. What we cannot know is what the trilobite made of that image. What neural processing happened behind those lenses? What the circuits translated the pattern of activation across thousands of facets into? Whatever the trilobite equivalent of a perception was, the fossil preserves the instrument. It does not preserve the experience. That gap between the optical hardware and the inner life it served is one of the deepest gaps in all of paleontology, and it is worth sitting with, because it applies not just to trilobites, but to every animal in this story, and eventually, in ways that become strange and uncomfortable when you press on them. To us. A trilobite with corrected calcite lenses was almost certainly seeing something. There was almost certainly something it was like to be that animal, looking out through its stone eyes at a Paleozoic sea full of things that wanted to eat it or that it wanted to eat. But that experience, whatever it was, ended 250 million years ago, and no fossil will ever give it back to us. What the fossils do give us is the trajectory, the arc of what was happening to vision across those millions of years. Eyes getting larger, eyes getting more complex, eyes appearing in new positions on new body plans. The record of trilobites is a record of vision being pushed and shaped and refined and diversified under constant ecological pressure. And then it stops. The Permian extinction, 252 million years ago, was the largest mass extinction in the history of animal life on Earth. Somewhere between 90 and 96% of all marine species disappeared. The causes are still debated, but the leading hypothesis involves massive volcanic eruptions in what is now Siberia, releasing enough carbon dioxide and sulfur dioxide to acidify the oceans, warm the climate, and deplete oxygen from the water over a period of tens of thousands of years. The trilobites did not survive it. After 270 million years, through ice ages and greenhouse periods and the rise of fish and the colonization of land, through every extinction event that had come before, the trilobites finally ran out of runway. Their stone eyes went dark. The lineages that survived the Permian extinction carried the lessons of 500 million years of visual evolution forward into a world that would, in time, be rebuilt from what remained. Eyes that had been refined under Cambrian pressure, tested through Devonian extinctions, diversified through Carboniferous forests, would now face a world with different animals, different pressures, different ecological relationships. But one thing that world would contain, in a cave system in Mexico, hundreds of millions of years later, was a discovery that nobody expected, a discovery that would force a complete rethinking of everything we thought we knew about why eyes exist at all. There is a river in northeastern Mexico called the Rio Choy, and it does something that rivers are not supposed to do. It goes underground. The water flows into a cave system in the Sierra de El Abra Mountains and continues underground for kilometers, through passages that have never seen sunlight, through chambers where the darkness is so complete that the concept of darkness barely captures it. This is not the darkness of a room with the lights off. It is not the darkness of a moonless night in the countryside. It is the physical condition of a place that light has never reached, absent human intervention, would never reach. In the pools and streams of that underground system lives a small silver fish, a few centimeters long, that belongs to the species Astyanax mexicanus. On the surface, Astyanax mexicanus looks like what it is, a small, ordinary, freshwater fish with functional eyes, normal coloration, and the standard visual behavior of a sighted animal. It responds to light. It navigates by sight. It does everything a fish with eyes is supposed to do. The cave version of the same species has no eyes. Not damaged eyes, not small or malformed eyes. The cave fish begins developing eyes in the embryo. The same genetic program initiates. The same early structures form. And then the process reverses. The developing eye degenerates before it is complete. What remains at birth is a small pocket of tissue under the skin where the eye would have been, with no functional optics, no photoreceptors capable of forming an image, no connection to any visual processing center in the brain. The fish is not blind in the way that a blinded fish is blind. It is blind in the way that a fish that has spent thousands of generations having its eyes taken away is blind. When researchers first established that the sighted surface fish and the blind cave fish were the same species, capable of interbreeding and producing viable offspring, it opened one of the most productive natural experiments in evolutionary biology. Because here was not just an example of an animal losing its eyes. Here was a system where you could study exactly how it happened and why. The why turns out to be more interesting than it first appears. The obvious explanation for why cave fish lose their eyes is that eyes are useless in complete darkness. So there is no selection pressure to maintain them. and they gradually degrade through the accumulation of random mutations that go uncorrected because they have no negative effect on survival. This explanation is true as far as it goes, but it is incomplete. It describes how the eyes could be lost without fully explaining why they are actively lost. Why? The process seems to be pushed rather than simply neglected. The more revealing explanation involves what the fish does with the resources it would otherwise spend on eyes. Eyes are expensive. The tissue is metabolically costly to build and maintain. The neural processing required to handle visual information takes up significant brain resources. In a sighted fish, this expenditure is justified by the return. Vision provides information that makes the animal more effective at finding food, avoiding predators, navigating its environment. The investment pays off. In complete and permanent darkness, it pays off nothing. The return on the investment drops to zero, while the cost remains exactly the same. What researchers found when they looked carefully at cave fish was that the resources not spent on eyes were being redistributed. The lateral line system, a network of pressure-sensitive organs running along the sides of the fish that detects vibrations and water movement, is significantly more developed in cave fish than in their surface relatives. The number of taste buds is dramatically higher, particularly around the mouth, allowing more effective detection of food particles in the water. The jaw structure changes in ways that make it better suited to finding food on the substrate. Where nutrients are more consistently available in cave environments, the cave fish is not a degraded version of the surface fish. It is a fish that has been re-optimized for a different world. Evolution did not simply allow eyes to decay through neglect. It actively redirected developmental resources from a costly and useless structure toward structures that provide genuine benefit in permanent darkness. This happens independently, in cave systems on multiple continents, in multiple unrelated species. Blind cavefish exist in Mexico, in Texas, in the limestone cave systems of Slovenia and Hungary, in the karst regions of Madagascar. In each case, the same logic applies. The same pressure produces the same outcome through different genetic mechanisms in different lineages. The cave fish of different continents did not lose their eyes because of some shared ancestor that went blind. They lost their eyes because darkness makes the same demand on every creature that lives in it. And the most effective response to that demand always looks similar, regardless of which animal is making it. This is the pivot point of the entire story. For 6 chapters, this has been a story about building eyes, about the molecular foundations of photoreception, about the physics of cups and pinholes and lenses, about the Cambrian arms race that turned vision into the central fact of animal ecology, about the extraordinary stone eyes of trilobites that preserved themselves in the rock for us to find and marvel at. The story has had a direction, toward complexity, toward sophistication, toward better and sharper and more capable. The blind cavefish disrupts that direction entirely. Evolution does not have a goal. It does not move toward complexity or sophistication or perfection or any other destination. It moves toward whatever keeps particular animals alive, in particular environments, at particular moments. When that means building extraordinary compound eyes with corrected calcite lenses, it builds them. When that means taking eyes away and redistributing the resources to taste buds and pressure sensors, it takes them away without hesitation and without any sense that something valuable is being lost. Eyes are not progress. They are tools. And like all tools, their value is entirely dependent on what they are being used for and whether the environment they are used in has any use for them at all. The trilobites had 270 million years of extraordinary eye evolution, and still went extinct when the chemistry of the ocean changed in ways their eyes could not see coming. The cave fish threw away eyes that had been refined over hundreds of millions of years of vertebrate evolution because the thing those eyes were designed to detect simply was not there anymore. Neither story is about success or failure in any deep sense. Both are about fit, about the match between what an animal has and what the environment demands of it. And this raises a question that the cave fish forces on anyone who thinks carefully about what they have been reading. If eyes can be built and rebuilt and taken away and rebuilt again as many times as the environment demands, how many times has the basic problem of vision been solved? Not across one lineage, not as a single invention that spread through the animal kingdom and was elaborated from there, but as genuinely independent solutions arrived at by different animals working with different materials in different parts of the world at different times. The answer, when researchers began trying to count, was not what anyone expected. In 1994, a Swedish biologist named Dan Erik Nilsson sat down to try to count how many times eyes had evolved. Not how many kinds of eyes existed. How many times? Independently. Starting from scratch, in lineages with no previous image-forming vision. Evolution had arrived at the ability to form an image from light. The number he arrived at was around 40. Not 40 variations on a single original design. Not 40 improvements to something a common ancestor had built. 40 separate origins. 40 times that natural selection. Working with whatever materials happened to be available in whatever animal happened to be under sufficient pressure, had independently solved the problem of turning light into a spatial map of the world. The number has been debated since then, as numbers in biology always are, because the definition of what counts as a true image-forming eye versus a more primitive light-sensing organ is not always clear. Some researchers put the number higher. Some argue for a more conservative count, but the broad conclusion is not seriously disputed. Eyes evolved many times, in many lineages, independently, because the problem they solve is so fundamental, and the pressure to solve it is so relentless that life keeps arriving at the same answer from different directions. And here is what makes it remarkable. Not just that this happened, but how different the answers are, because the physics of light has only a limited number of solutions. You might expect that every independently evolved eye would converge on the same basic design. Camera lens plus retina plus brain. The way aerodynamics produces similar shapes in unrelated flying animals. But that is not what happened. Instead, life found ways of using light that human engineers had never considered. Using materials that engineers would never have specified. Producing visual systems that process information in ways that reveal how narrow our intuitions about vision really are. Start with a scallop. A scallop is not an animal that most people associate with sophisticated sensory biology. It is something you order in a restaurant or find on a beach. But run your finger slowly along the edge of a living scallop's mantle, the soft tissue that lines the inside of the shell, and you will see something that stops most people cold the first time they notice it. Rows of tiny bright blue eyes, dozens of them, sometimes more than 100. Each one a complete visual organ with a cornea, a lens, and a retina, all contained in a structure only a millimeter or two across. And not one of them uses a lens to focus light. They use mirrors. Behind the retina of each scallop eye is a layer of tiny crystals arranged in a precise mosaic, forming a curved reflective surface that works like a concave mirror, gathering light and focusing it back onto the retina the way a reflecting telescope gathers and focuses starlight. This is how the Hubble Space Telescope works. and how the James Webb Space Telescope works. Not by bending light through glass, but by bouncing it off a precisely curved mirror. Scallops have been using the same optical principle for hundreds of millions of years, in an eye the size of a pinhead, to detect the shadow of an approaching starfish and trigger the escape response of snapping the shell shut and jetting away through the water. The scallop does not need to identify its predator. It does not need to read a face or track a moving target across a complex background. It needs to know that something large is moving toward it from some direction. 100 mirror eyes, each watching a different patch of space, are a nearly perfect solution to that specific problem. Now, consider the box jellyfish. Box jellyfish, the group that includes some of the most venomous animals on Earth, present a problem that has occupied researchers for decades. They are related to ordinary jellyfish, animals with a famously simple body plan and no centralized brain in any conventional sense. The nervous system of a jellyfish is a diffuse net of neurons spread through the body, without the kind of central processing structure that we associate with complex behavior. Box jellyfish have 24 eyes, not simple photoreceptor patches. 24 eyes organized into 4 clusters of 6. Each cluster called a rhopalium, and each rhopalium containing eyes of 4 different types. 2 of those types are camera-type eyes with a cornea, a spherical lens, and a retina, capable of forming real images. They are structurally similar to the eyes of vertebrates in ways that are almost certainly the result of convergent evolution rather than shared ancestry. A jellyfish with camera eyes and no centralized brain. The upper lens eyes of some box jellyfish species are permanently oriented upward, maintained in that position by a weighted crystal structure that acts like a gyroscope, keeping the eye pointing at the sky regardless of how the jellyfish itself is oriented as it pulses through the water. Behavioral studies suggest that box jellyfish use this upward gaze to navigate by the visual canopy above them, keeping themselves oriented within mangrove environments by tracking overhead light patterns. In murky, tropical water, where chemical and pressure cues are unreliable, visual navigation by sky pattern may be the most consistent guide available. An animal with no centralized brain uses camera eyes to navigate by reading the sky. Now consider the mantis shrimp, which has become something of a celebrity in popular science writing, though usually for the wrong reasons. Mantis shrimp have 16 types of photoreceptors. The internet has cheerfully informed millions of people that mantis shrimp therefore see color 16 times better than we do, or experience a visual world of incomprehensible richness and complexity compared to our own. This is not what the science actually shows. Behavioral experiments testing the ability of mantis shrimp to discriminate between colors show that they are actually worse at distinguishing between closely similar colors than humans are. A mantis shrimp cannot make the fine color distinctions that a human can make with only 3 cone types. What the 16 photoreceptor types appear to do instead is something more interesting and more alien. Rather than comparing signals across receptor types to determine color the way our visual system does, mantis shrimp seem to use each receptor type as a separate labeled channel. When a specific wavelength of light hits a specific receptor, that receptor fires, and the signal means something specific without needing to be compared to anything else. It is less like mixing colors on a palette and more like reading a barcode. Each position on the barcode either activates or it does not. This system is faster than comparative color analysis. It requires less neural computation. It is not better at distinguishing similar colors, but it is faster at categorizing objects that reflect specific wavelengths. which matters enormously if you are a mantis shrimp whose survival depends on identifying prey and committing to a strike in the fraction of a second available before the moment passes. The mantis shrimp visual system is not a better version of human color vision. It is a completely different approach to the same problem of extracting useful information from light, optimized for a completely different set of priorities. And finally, before we leave this catalogue of invented eyes, consider the chitin. A chitin is a marine mollusk that clings to rocks in the intertidal zone. A flattened oval creature with a shell made of 8 overlapping plates. Some chiton species have eyes embedded directly in those shell plates, not in soft tissue adjacent to the shell, but in the mineral structure of the shell itself. The plates of these chitons contain small transparent structures made of aragonite, a form of calcium carbonate, which function as lenses. The chitin sees through its own armor. Both chitons and trilobites built optical structures from minerals, from different forms of the same basic compound, calcium carbonate, arrived at independently in lineages with no close common ancestor. Different materials, different structures, different geological periods. The same fundamental approach to the same fundamental problem. Here is what all 4 of these animals share with each other and with the 40 or so other independent origins of image-forming vision. None of them built the eye that a human engineer would have designed. None of them used the materials that optical engineering would specify. None of them produced a system that looks, from the outside, like the obvious solution to the problem they were solving. Each of them found a path through the space of physical possibilities that was constrained by what materials their lineage happened to have available. What developmental programs existed that could be co-opted, what ecological pressures they were under, and what incremental improvements happened to be accessible at each step. The result is not a collection of variations on a theme. It is a collection of genuinely different answers to the question of how to extract useful information from light. And 40 different answers to the same question raises something worth pausing over before this story moves forward. If the scallop and the box jellyfish and the mantis shrimp each built different eyes that perceive light in different ways, are they living in the same world? You have probably had this experience. You are walking through a garden or a park in summer, and you notice a bumblebee moving from flower to flower. It seems to have preferences. It bypasses some flowers entirely and goes directly to others. And if you look carefully at the flowers it chooses versus the ones it ignores, you cannot always see an obvious difference. The colors look similar to you. The shapes look similar. But the bee knows something you do not. What the bee knows is written in ultraviolet light. The petals of many flowers reflect ultraviolet wavelengths strongly, in patterns that are completely invisible to human eyes, because our photoreceptors do not respond to those wavelengths. To a bee, whose eyes contain ultraviolet-sensitive photoreceptors, those flowers are not the relatively uniform colored objects you see. They are objects with visible markings, lines and patches and bullseye patterns centered on the nectar-containing structures, a landing guide as clear and specific as runway lights at an airport. Flowers did not evolve colors because they are beautiful. They evolved colors as communication with pollinators whose eyes were sensitive to specific wavelengths. The beauty that humans perceive in flowers is a side effect of a signaling system that was not designed for human perception at all. You happen to have photoreceptors that respond to some of the relevant wavelengths. So you see color in flowers, but you are receiving a partial and distorted version of a signal that makes full sense only to the eyes it was built for. The bee in that garden and you in that garden are standing in the same physical location. You are breathing the same air, feeling the same temperature, surrounded by the same flowers, and you are not in the same world. This idea that different animals inhabiting the same physical space nonetheless live in genuinely different sensory worlds was developed most carefully by a Baltic German biologist named Jakob von Uexküll in the early part of the 20th century. Von Uexküll called each animal's subjective sensory world its Umwelt, a German word meaning roughly surrounding world. His point was not mystical, it was biological. Every animal's sensory systems extract a specific subset of the information available in the physical environment, the subset that matters for that animal's survival. And that subset defines the world the animal actually lives in. The physical world is the same for everyone in it. The Umwelt is not. Walk into that same garden at night and you enter a world that belongs to different animals entirely. A moth navigating in darkness is using a sensory world built primarily from scent and from the pale colors of night-blooming flowers. A bat moving through the same space is building a real-time three-dimensional map from the echoes of its own ultrasonic calls. You, standing in that garden at night, are largely blind to both of those worlds. Consider what a migratory bird experiences during its annual journey of thousands of kilometers across open ocean. Evidence now strongly suggests that some birds can detect magnetic fields, and that this detection may involve the visual system directly. Through light-dependent reactions in photoreceptor proteins that produce signals influenced by the orientation of the Earth's magnetic field. The exact mechanism is still being worked out. The subjective experience of the bird, if there is one, cannot be known from the outside. But the magnetic information appears to be processed in brain regions associated with vision, suggesting that for some birds, magnetic orientation may be experienced as something integrated with what they see. Not a separate feeling, but something closer to a visible cue overlaid on the world around them. A warbler crossing the Gulf of Mexico at night may be navigating by a combination of star patterns and something that functions, in some sense, as a compass written into the sky itself. Or consider a rattlesnake hunting in complete darkness. The pit organs on either side of a rattlesnake's face are not eyes. They do not form images through refraction or reflection. They detect infrared radiation, heat, emitted by warm-blooded prey animals. The neural pathways from these organs connect to the same brain region that processes visual information from the eyes. The result is something that functions as a heat image overlaid on whatever visual information is available. A second representation of the world, built from temperature rather than light, telling the snake where the warm things are in the darkness. The snake has two visual worlds running simultaneously, one built from light, one built from heat. What all of these animals share is a common situation. Each of them lives in a physical environment that contains far more information than any single sensory system can capture. And each of them has evolved a system that captures the specific slice of that information that matters most for its survival. Your visual system captures a specific range of electromagnetic wavelengths, roughly between 400 and 700 nanometers, that your evolutionary history has made relevant. You do not see ultraviolet or infrared or magnetic fields or polarization patterns, not because those things are not there, but because the specific pressures that shaped your lineage never made detecting them a matter of survival. The physical world is one thing, the world that any given animal actually inhabits, the world defined by what its senses can reach and what its nervous system can make meaningful, is something smaller and more specific and, in a certain sense, more personal. And here is what this means for the question of how many worlds exist right now. in the garden where the bee sees ultraviolet landing guides on flowers, and the moth navigates by pale petals and night-intensified scent, and a nearby rattlesnake detects the warmth of a mouse moving through the grass, and a migrating warbler overhead navigates by stars and possibly by something integrated into its visual field that reads the Earth's magnetic orientation, and you stand looking at what you would describe as a pleasant summer evening. Every one of those animals is in the same physical location. None of them is in the same world. The worlds do not communicate. They do not overlap in any meaningful experiential sense. They occupy the same coordinates in space and time and share almost nothing of what it is like to be inside them. How many of those worlds are there? As many as there are sensory systems, as many as there are different solutions to the problem of extracting useful information from a physical environment. And as the previous chapter showed, when it comes to vision alone, that number is at least in the dozens, operating right now in every environment on Earth. But among all of those worlds, built by all of those eyes, using all of those different approaches to the physics of light, there is one that stands apart from the others in a specific and unexpected way. It stands apart not because it sees more wavelengths, or processes color better, or has higher resolution, or covers a wider field, or works in lower light. By most of those metrics, it is outperformed by other animals. It stands apart because of what it looks for above everything else. And what it looks for, more than food or predators or ultraviolet patterns on flowers or magnetic fields written in the sky, is a face. Look at a photograph of a chimpanzee's face. Look specifically at the eyes. The iris is dark, often almost as dark as the pupil, and the area around it. What would be the white of the eye in a human is heavily pigmented. In most lighting conditions, you cannot easily tell where a chimpanzee is looking unless you are very close and watching carefully. The direction of its gaze is hidden. The eyes do not broadcast where attention is going. Now look in a mirror. The white of your eye, the sclera, is exposed on either side of the iris across a horizontal band that is unusually wide compared to any other primate. When you shift your gaze to the left, the iris moves left, and the white space on the right side increases visibly. When you look up, the white below the iris expands. The direction of your attention is readable from across a room. Anyone watching you can tell where you are looking without needing to follow your head movement or get close enough to study your face. This is strange when you think about it from a survival perspective. For most animals, concealing the direction of gaze is advantageous. A predator that makes its hunting intention obvious by broadcasting where it is looking gives prey time to react. An animal navigating a complex social environment benefits from some ambiguity about what it is attending to. Visible gaze, direction is information, and information handed freely to others is a vulnerability. In humans, the evolutionary pressure seems to have run in the opposite direction. The exposed white sclera of the human eye is not an accident or a byproduct of some unrelated change. Comparative studies across primates show that humans have a significantly larger exposed scleral area than chimpanzees, gorillas, orangutans, or any other great ape. The difference is structural, consistent, and appears to be a derived feature of the human lineage rather than a shared ancestral trait. One influential hypothesis, called the cooperative eye hypothesis, developed by researchers including Michael Tomasello and colleagues in the early 2000s, proposes that visible gaze direction in humans evolved to facilitate the kind of joint attention that is essential for cooperative behavior. If you and I are working together on a task, and I need to direct your attention to something specific without making noise or moving dramatically. I can look at it. You can follow my gaze. We have communicated without speaking. This matters more than it might seem. Joint attention. The ability to coordinate attention with another individual toward a shared object or location is a capacity that underlies a remarkable range of specifically human behaviors. Teaching requires one individual to track what another individual is and is not attending to. Pointing and following points requires understanding that another individual's gaze or gesture is meaningful and directed. Cooperative hunting, where multiple individuals need to coordinate their attention to a moving target without vocalizing in ways that would alert it, depends on being able to read where others are looking in real time. Chimpanzees, for all their intelligence, are much less reliable at following the gaze of another individual and much less likely to use their own gaze to direct attention in the flexible and intentional way that human children do from a very early age. The difference correlates with the difference in scleral visibility. The white of the eye is a communication channel, broadcasting attentional state to the people around you. And humans have a dramatically more visible version of that channel than any of our closest relatives. The cooperative eye hypothesis has support from comparative anatomy and from behavioral studies. Though researchers continue to examine and debate the precise mechanisms and the range of explanations for scleral coloration differences across primates, what is not in dispute is the anatomical fact human eyes broadcast gaze direction in a way that is unusual in the primate world. Your eyes are not just organs for receiving information about the world. They are organs for transmitting information about your internal state to the people around you. And they are extraordinarily good at it. The human fovea, the high-resolution central region of the retina, is positioned and sized in a way that makes it particularly well-suited to reading fine detail at the distances typical of face-to-face social interaction. When you look at another person's face, your eyes move in a characteristic pattern. Rapid movements, called saccades, that reposition the fovea dozens of times per second, sampling the eyes, the mouth, the nose, then back to the eyes, in a sequence that is recognizable across cultures and appears very early in development. You are not passively receiving a face. You are actively reading it, allocating your highest resolution sampling to the features most likely to carry meaningful social information. The brain structures devoted to processing faces reflect how central this task is to human visual life. A region of the temporal lobe, called the fusiform face area, responds selectively and powerfully to faces, including faces seen only briefly, faces partially obscured, and faces of people never met before. Damage to this region produces a condition called prosopagnosia, the inability to recognize faces, which can be so severe that affected individuals cannot recognize their own family members from their faces alone. Even with intact vision and intact recognition of every other category of object in the world, faces are not just one category among many in human visual processing. They are a privileged category, with dedicated neural machinery, with processing that begins faster and operates more automatically than almost any other visual stimulus, and with an importance to social function that has no parallel in our sensory lives. Research with newborns. Building on foundational work, including studies by Goran, Sarti, and Wu, has found that infants in the first hour of life show a preference for tracking face-like patterns over other visual stimuli of similar complexity. The precise mechanisms behind this early preference are still being studied and debated, but the basic observation has been replicated enough times to carry weight. Something in the newborn visual system responds preferentially to the configuration of features that faces share. Before the child has had any experience that could teach it what a face means. The preference is there before the knowledge. The eye was oriented toward faces before the brain knew what faces were for. Think about what this means for what human vision is actually built to do. In the long history of vision, each improvement served a clear and immediate survival function. Better spatial resolution meant better detection of distant predators. Faster motion processing meant quicker escape responses. Color vision in primates appears to have been particularly useful for identifying ripe fruit against a background of foliage. The connection between visual capability and survival outcome was direct and relatively simple to trace. The human eye's orientation toward faces serves survival functions too. Reading threat in another person's expression, tracking the attention of group members during cooperative tasks, identifying individuals across a community of dozens or hundreds of people who need to be remembered and treated differently based on complex histories of interaction. These are survival-relevant capabilities in a deeply social species, but they are not the same kind of survival as detecting a predator or finding food. They are social survival. Survival in a world where your most important relationships are with other members of your own species, where your success depends not just on your own physical capabilities, but on your ability to cooperate with and read and be understood by the people around you. The scallop needs to know if a shadow is approaching from any direction. The mantis shrimp needs to categorize what it is about to strike in the fraction of a second available. The migrating warbler needs to know which way is south. The cave fish needs to find food in permanent darkness. The human needs to know what another human is thinking. And so, the eye that evolution built for us, with its wide, white, sclera broadcasting gaze direction, its fovea positioned for reading the fine details of an expression, its neural processing prioritizing faces above almost every other visual stimulus, is not the eye that would win a competition for raw acuity, or field of view, or color range, or low light sensitivity. It is the eye that is best at the one thing our survival has always depended on more than anything else: reading the person in front of you. Try something right now. Hold up your right hand with the thumb pointing upward. Close your left eye. Look straight ahead with your right eye, fixing your gaze on a point directly in front of you. Do not let your eye wander. Now slowly move your thumb to the right, keeping it at arm's length, keeping your gaze fixed straight ahead. At some point, your thumb will disappear. Not fade, not blur, disappear completely. There will be an empty space where your thumb was, and what fills that space will be whatever is behind your thumb. The wall, a window, the sky, rendered continuously and seamlessly as though your thumb was never there. Move your thumb a little further and it reappears. Move it back and it vanishes again. You have just found your blind spot. Every human eye has one, in exactly the same location, for exactly the same reason. The optic nerve, the bundle of roughly 1 million nerve fibers carrying visual signals from the retina to the brain, has to exit the eye somewhere. Where it exits, there are no photoreceptors. There is nothing to detect light. If an image falls on that spot, it is simply not registered. The size of this gap in your visual field is not trivial. At arm's length, objects roughly the size of your thumbnail fall entirely within your blind spot and are completely invisible to you. You have had this hole in your vision your entire life. You have almost certainly never noticed it. Not because you have not looked, but because your brain fills it in so automatically, and so completely, and so immediately, that the filling happens before anything reaches conscious experience. The brain looks at the information surrounding the gap, estimates what should be there based on what it finds around the edges, and inserts that estimate into your visual experience with no seam and no signal that anything unusual has occurred. The blind spot is the most visible example of something the brain does constantly, in every part of your visual field. At every moment, you are awake. Your eye sends the brain incomplete information. It cannot do otherwise. The retina has gaps in its receptor coverage. The fovea vert, the only region of genuinely high resolution, covers only a tiny fraction of your total visual field. The eye moves constantly, which means the image on the retina is always shifting and always being interrupted. Light levels change. Objects move in and out of shadow. The two eyes send slightly different images that must be reconciled into a single coherent perception. The brain does not display what the eye sends. It builds a model. This is not a loose metaphor. It is a description of what the neural processing actually does. Visual information arriving from the retina travels through multiple processing stages in the brain, and at each stage, the raw input is combined with predictions, with context, with memory of what similar inputs have meant before, to produce a representation that is more stable, more coherent, and more useful than the raw sensory signal alone. Every time your eyes move, which happens 3 to 5 times per second when you are looking around a scene, the visual signal from the retina is briefly disrupted by the motion itself. If the brain displayed that disruption, your visual world would smear and blur several times every second. Instead, visual sensitivity is briefly reduced during each eye movement, and the brain stitches together the samples taken between movements into a seamless experience of a stable and continuous visual world. You are not seeing continuously. You are seeing a series of snapshots assembled by a brain that is very good at hiding the gaps between them. The consequences of this become visible in predictable ways. In experimental settings, researchers can make large and obvious changes to visual scenes during brief interruptions. The flicker of a blank frame in a video, the moment when an observer blinks, a distraction that pulls attention away for a fraction of a second. Objects change color. People switch clothing. An entirely different person walks into the frame in place of the one who was there before. Observer, watching carefully and knowing that changes are occurring. Miss them with surprising frequency. The brain, predicting a stable world, does not always register when the world has changed. And then there is color constancy. The color you perceive an object to be is not determined solely by the wavelengths of light it actually reflects into your eye. It is determined by those wavelengths after the brain has adjusted for its estimate of the ambient illumination. A white piece of paper reflects short wavelengths strongly in fluorescent light and long wavelengths strongly in candlelight. The brain adjusts for the illuminant and sees white in both cases, because white is what the paper is, and white is what the brain will represent it as under reasonable lighting conditions. This system usually works beautifully, but occasionally the cues the brain uses to estimate illumination are ambiguous. When that happens, different people's brains make different estimates, and they see genuinely different colors in the same object. This is what happened with a photograph of a dress that circulated widely in February of 2015. The dress was photographed under lighting conditions that made the illuminant ambiguous. Some brains estimated one illuminant and saw blue and black. Other brains estimated a different illuminant and saw white and gold. Both perceptions were the output of visual systems working exactly as they were designed to work. The systems simply made different assumptions about the context. Same photograph, different realities. The visual world you experience feels direct. It feels like a transparent window onto what is actually there. The colors look like the colors of things, not like a brain's estimate of the colors of things. The seamless, stable, richly detailed world you navigate through your day feels given, not constructed. It is constructed, every moment of it. This is not a flaw. The construction is the feature, a visual system that showed you the raw input, with its gaps and its motion blur and its illumination shifts and its retinal inconsistencies, would be harder to act on, not easier. The construction smooths all of that away and delivers something coherent and actionable. For the purposes of moving through the world and responding to what is in it, the model is better than the raw data. But the model is not the world, and the gap between the model and the world is not always trivial. Here is where Michael Land and the question from the prologue become relevant again. When Land looked at the human eye as an optical instrument and noted all of its apparent flaws, he was right about the optics, the inverted retina, the blind spot, the uneven resolution. These are genuine optical limitations. But the error in treating the eye as a standalone device is the assumption that it is supposed to produce output the way a camera produces output, as an image that stands on its own. The eye does not produce output that stands on its own. The eye is part of a system, and the other part of that system is a brain that has spent hundreds of millions of years learning how to compensate for precisely those optical limitations. The inverted retina is not a mistake because the brain's processing handles what that arrangement produces. The blind spot is not a problem because the brain fills it in. The low resolution of the periphery is not a liability because the brain integrates high-speed eye movements to sample the scene comprehensively. The eye and the brain are not two components that happen to be connected. They are a single system that evolved together, each shaped by the other, each making assumptions about what the other will do. You cannot evaluate one without the other. What the whole system produces, after 500 million years of refinement, is not a perfect image of the world. It is something more useful than that. A world you can live in. The delivery room is bright. Hospitals are always bright. The kind of flat, institutional light that fills every corner and casts almost no shadow to the doctors and nurses working in it. This light is functional and unremarkable, the background condition of a workplace they have stood in hundreds of times, to the person who has just arrived in it. After 9 months of complete darkness, it is something else entirely. A newborn baby, in the first minutes after birth, has a visual system that is present but not yet operational in the way it will be 6 months from now, or 2 years from now, or 40 years from now. The structures are there. The retinas contain photoreceptors. The optic nerves are connected to the visual cortex at the back of the brain. The eyes open, and they respond to light, the pupils constricting in the brightness of the delivery room. But what the baby sees, in those first minutes, is not what you see when you look at the same room. The fova, the high-resolution central region of the retina that allows fine detail vision, is not yet fully developed at birth. The myelin sheathing on the optic nerve fibers, the insulating layer that allows signals to travel quickly and without degradation, is incomplete. The visual cortex itself is present but largely unactivated by experience. Its circuits not yet shaped by the input that will, over the coming months, organize them into the efficient processing architecture of an adult visual system. What the newborn receives is high contrast at edges, where light meets dark, large shapes moving against backgrounds, and the general fact of illumination. The rich, detailed, colorful, depth-structured world that a sighted adult experiences is not available. What is available is something much simpler, and, in a specific historical sense, something much older. The newborn's visual world, in those first minutes, is closer to what the earliest image-forming eyes in the Cambrian Ocean would have provided than to what the same child will experience at age 10. This is not because development literally replays the history of evolution. It does not, but there is a parallel worth noticing. Vision begins in development, as in evolutionary history, not with richness and detail, but with the most basic information that light can carry: contrast, motion, the difference between bright and dark. From that beginning, the development of human vision follows a sequence that researchers have mapped in enough detail to trace week by week and month by month. In the first weeks, the visual system responds most strongly to high contrast patterns and to movement. A face at close range, around 20 to 30 centimeters, which is roughly the distance from a nursing infant to the face of the person holding it, registers as a pattern of light and dark regions. The eyes, the mouth, the hairline, these are visible as contrasts. The face as a meaningful object, as a specific person with a history and a name and intentions that matter, is not yet there. Around 2 months, infants begin tracking moving objects more smoothly, following them with sustained gaze rather than the jerky catches and losses of the newborn period. The system that keeps a moving object on the fovea as it crosses the visual field comes online. The world has started moving in a way that can be followed. Color vision develops across the first months, with sensitivity to longer wavelengths appearing before sensitivity to shorter wavelengths. The full complement of cone types is present from birth, but the neural processing that interprets their signals matures gradually, somewhere between 2 and 4 months. Something shifts in how infants respond to faces. Studies using eye-tracking technology show that infants in this age range begin to scan faces in the pattern characteristic of adult face processing, prioritizing the eye region, moving to the mouth, returning to the eyes. The brain is beginning to organize itself around the task that the social eye was built for. Faces are becoming special. Around 4 to 6 months, stereoscopic depth perception comes online, as the visual cortex learns to interpret the slight differences between the images received by the two eyes as information about the distance of objects. The world acquires a third dimension in the infant's experience in a way that it did not fully have before. By the end of the first year, the visual system is processing faces, tracking motion, perceiving color and depth, and integrating all of this with information from the other senses and from the infant's own movements through space. The architecture that will serve the adult for decades is substantially in place. What happened in those 12 months is not a miracle or a mystery, though it is genuinely extraordinary. A system of almost unimaginable complexity assembled itself, guided by a genetic program that laid down the basic wiring and then shaped by the experience of actually using that system, of receiving visual input and organizing neural responses to it, of building the predictive models that will eventually run so automatically that the adult will have no conscious sense of effort in seeing at all. But go back to the delivery room. Go back to the first 8 minutes. The baby's eyes are open. The pupils are responding to the brightness. The visual system is receiving its first sustained input from the world outside. Research going back decades, including influential work building on foundational studies by Gorin, Sati, and Wu, has found that very young infants, including infants measured within the first hour of life, show a preference for looking at patterns that resemble faces over patterns of similar complexity that do not. The finding has been examined, replicated, questioned, refined, and examined again across many laboratories and many years. The precise mechanisms underlying this early preference are still being worked out by developmental researchers. What the data consistently shows is that the preference is there early, earlier than learning could have produced it, earlier than any face has been studied long enough to be recognized as familiar. The newborn visual system arrives in the world already oriented, in some functional sense, toward the thing that the social eye was built over millions of years to find. Now look at what is happening on the other side of that exchange. The mother who looks into the eyes of her newborn in those first minutes is carrying a visual system built by the same 500 million years of evolutionary history. Her fusiform face area is firing. Her gaze direction system is broadcasting toward the infant. Her brain is building a model of the face in front of her, fitting it into a category that has no parallel in human neural processing. A face that is new, but that carries a weight that no other new face ever will. She sees an infant who appears to look back at her, who appears to track the movement of her face, who appears, against all the odds of an incomplete and largely unorganized visual system, to be present in some way that she recognizes before she has words for it. What the infant sees is harder to say. The face of the mother, at the distance of nursing, is within the range where even an underdeveloped fovea can register a high contrast pattern. The dark pupils against lighter irises, the boundary of the hairline, the movement of a mouth. These are exactly the kinds of signals that the newborn visual system is best equipped to detect. Whether any of this constitutes seeing a face in the way an adult sees a face, whether there is recognition or experience or anything that would be called perception in any rich sense, is a question that cannot be answered from the outside. But the face is there, and the eyes are open, and the system that spent 500 million years learning to extract useful information from light in its earliest and most incomplete form, doing what it was built to do, looking. Michael Land eventually stopped trying to understand the human eye as an optical instrument, not because the project was uninteresting, because it was incomplete. After years of analyzing the eye's apparent flaws, the inverted retina, the blind spot, the uneven resolution, the way the whole system depended so heavily on a brain compensating for what the optics could not do, he arrived at a conclusion that reframed the original question entirely. The human eye was never built to be a camera. It was built to be part of a system. And the system it was built to be part of was not an optical system. It was a social one. An eye and a brain and a body moving through a world full of other eyes and brains and bodies. All of them reading each other. All of them transmitting and receiving information that no camera has ever been designed to capture. He wrote about this. He continued researching. He became one of the most influential figures in the study of animal vision across the second half of the 20th century and into the 21st, producing work on eyes so different from each other that they sometimes seem to belong to different universes of biology. And at some point during those years, he noticed something he could not account for. He was in a museum, standing in front of a painting he had walked past many times without stopping. On this particular day, he stopped. He does not record exactly what made him stop that day, rather than any other. Something about the quality of the light in the painting. The way the artist had rendered the transition from brightness to shadow across a particular surface caught him in a way that had nothing to do with optical analysis. He stood there for several minutes, not examining, not cataloguing, not doing anything that could be described as gathering information for any purpose. Just looking. He thought about this afterward, because it is the kind of thing he thinks about. His visual system, built by 500 million years of selection pressure aimed entirely at survival, had stopped him in front of a rectangle of pigment on canvas and held him there for minutes, producing an experience that had no obvious survival value. No food, no threat, no social information directly relevant to anything he needed to do. A dead artist's interpretation of how light falls on a surface. And his eye, the badly designed camera, with the inverted retina and the blind spot and the uneven resolution, was doing something with it that he could not account for in any strictly functional terms. The eye was never built to find a painting beautiful. There is no pathway from Cambrian predator pressure to the experience of being unable to walk away from a painting. There is no selection pressure that specifically favored the capacity to be stopped by the quality of light in a landscape, or by the expression on a face rendered in oil on canvas by someone who died 300 years ago. The eye was built to detect edges and motion and color and faces, to gather information about a world full of things that could kill you or feed you or cooperate with you, to serve survival in a world that had no museums and no paintings and no concept of beauty as anything separable from health or food or the fitness of a potential mate. And yet here is land, stopped in front of a painting. Here is what the eye was built to do, laid out plainly. It was built to detect light and turn it into spatial information. It was built to find predators before they found you. It was built to locate food. It was built, in the specific case of human eyes, to read faces and track gaze and participate in the social processing that a deeply cooperative species requires. It was built to serve survival, in a world where survival was the central problem and beauty and meaning and wonder were not categories that had any independent relevance to whether you lived or died. Here is what it does, in the humans who carry it. It stops people in front of paintings. It watches light change on water for no reason except that the watching produces something that cannot be named precisely, but is not nothing. It looks up at a night sky and does something with the information it receives that goes considerably beyond locating objects in space. Something that led humans to name the stars, to trace patterns across them, to build entire systems of meaning from the arrangement of points of light that have no inherent arrangement at all. It finds, in the face of a stranger on a crowded street, something that lasts past the moment of seeing, an expression caught in passing that stays present in memory without being asked to. None of that is what the eye was built for. All of it is what the eye does. Go back to the delivery room one more time. The baby who opened its eyes 8 minutes after birth and oriented toward the high contrast pattern of its mother's face was doing the oldest thing that eyes do, turning toward the light, finding in the pattern of photons arriving from a specific direction something that the system, unfinished and unmyelinated and largely unorganized, already knew was worth attending to. That is what the first light-sensitive patch of cells in the Cambrian Ocean did, 500 million years ago. Turned toward the light, found in the direction of brightness something worth moving toward. The distance between that patch of cells and a human eye looking at a painting in a museum is almost impossible to hold in mind at once. 500 million years of building and rebuilding and discarding and starting over. 40 independent inventions of image-forming vision. calcite lens in trilobites, mirror eyes in scallops, camera eyes in jellyfish with no centralized brain, a fish in a Mexican cave slowly losing its eyes over thousands of generations because the light was gone and the investment was no longer worth making, a mantis shrimp processing color faster than any comparative analysis could manage, a newborn orienting toward the face it cannot yet fully see. All of it, every step, driven by the same pressure. The world is out there. You need to know what is in it. And somewhere in the long middle of all of that, in a lineage that climbed out of the ocean and walked upright and developed language and lived long enough to have time that was not consumed entirely by survival, the eye that was built to gather information about a dangerous world started doing something that the world it was built for never required of it. It started finding things worth looking at, not because that was the plan, because a system built with enough complexity to navigate a dangerous world, placed inside a brain with enough capacity to build models of that world and hold them and examine them, turns out to have capabilities that go beyond the ones it was assembled for. Land never did find a fully satisfying explanation for why he stopped in front of that painting. He has frameworks for why the visual system responds to certain kinds of contrast and certain kinds of light distribution. And some of those frameworks are relevant to what paintings do when they work. But the frameworks do not fully account for the several minutes of standing there, not analyzing, just present to something that had no survival relevance and was not nothing. He has decided, for now, to leave that gap open. Some of the questions the eye raises have answers in the 500 million years of history that built it. Some of them are questions about what the eye turned out to be.